everyone, I'm Juliette Smith. I'm an associate professor at the Virginia Institute of Marine Science. I'm in the Department of Aquatic Health Sciences. Today I'll be telling you about various projects within my research group that are all focused on harmful algal blooms and their toxins. My group therefore contains biologists, chemists, and toxicologists. Some of our research is focused in Chesapeake Bay, as well as other regions of the East, West, and Gulf coastlines of the US. My hope is that by the end of this presentation, you'll have a good idea of how my group researches harmful algal blooms and how we strive to produce data that are advancing the science, but also useful to resource managers and health officials in the protection of environmental and human health. So let's get acquainted with algae first. To start us off, phytoplankton are microalgae that use chlorophyll to harness light from the sun to grow. This is similar to plants doing photosynthesis on land. So, if you took a drop of water from your favorite bay, lake, pond, or ocean, dropped it onto a glass slide, and put it under a light microscope, you'd likely see a collection of living microscopic algae swimming about. Now, what we can do in the lab is pull out a single harmful algal cell from that water droplet and grow it up in a flask in the lab so we can study it using culturing experiments. So we can learn things about what makes that algal cell grow and produce toxins. And in the video on the bottom right, you can actually see two algal cells feeding on another round algal cell in the middle. So in this case, a harmful algal cell is actually feeding along with photosynthesis, making it a mixotrope. So now that we understand algae, what is an algal bloom? Algal blooms are the rapid growth of algae in the water, typically over 1,000 cells per mil. So in less than a quarter of a teaspoon of seawater, you'd have more than 1,000 cells of the same algae. But before we go any further, it's important to point out that algal blooms can be very beneficial to the aquatic environment. Energy produced by phytoplankton or algal blooms provides energy to higher trophic levels like our commercial fish and our marine mammals. But an algal bloom becomes harmful when the rapid growth of algae in the water, typically 1,000 cells per mil, has a negative impact on the environment, economy, society, and/or human health. Harmful algal blooms can contribute to degraded water quality when the harmful algal bloom accumulates as a scum on the surface or falls to the bottom of the water body and decomposes. So more specifically, HABs can lead to impacts such as light limitation, depletion of oxygen needed by fish and other marine organisms, coastal acidification, or the unpleasant odor, taste, and appearance that you can imagine would come with a rotting bed of algae. In my research group, we study another effect of HABs, the impact of algal toxins on organisms, and the accumulation of toxins in their tissues. So, in general, my group focuses on two major topics. First is the HAB ecology. This is the interaction of harmful algae with their environment. And second is the production of toxins by some of these harmful algae. So first we study the drivers of HABs, or things in the environment like nutrients or temperature that could cause HABs to grow and produce toxins. And second, we study the impacts of these toxins, including the toxic effect in aquatic animals, accumulation of the toxins in the food web, and then how long these toxins remain in the environment long after the bloom is gone. We use tools to conduct our science. On the bottom left, the imaging flow cytobot, or we call it our cytobot, is a robot that we can put into the water, and it acts as an underwater microscope, telling us what HABs are present and at what cell concentrations. We can use this tool to provide early warning to those in the bay of a developing HAB. We can also deploy a water quality sonde, or a multi-probe, which is the picture to the right, to measure water quality. Now, up in the top right, to investigate impacts of toxins, we rely on our chemistry. And we use a mass spectrometer to quantify toxins in the environment. I thought you might want to see some images of HAB scientists at work. So in the field, we deploy oysters or other bivalves in cages to determine the rates of toxin accumulation or detoxification in edible meat. And in the middle is an image of VIMS deploying our imaging flow cytobot and a water quality multi-probe. We also collect, filter, and sieve a lot of water in HAB science. We are also very active in the lab. We culture the algae in incubators, and we extract the toxins so we can measure them using mass spectrometry. So now, thinking back to our two main research topics, our research into drivers of HABs is one, working towards predicting blooms or providing that early warning. Mitigating, you can think of this as dealing with blooms when they occur, and hopefully someday, maybe even minimizing or preventing blooms. Our research into the impacts of algal toxins has the goals of securing seafood safety or understanding the rates of toxin accumulation in our edible seafood products. And thirdly, ensuring environmental health by understanding the persistence of toxins in the environment and effects on aquatic animals. And then fourth, our goals are contributing to aquaculture sustainability. So as we investigate ways to reduce the breakthrough of cells and toxins into facilities, and we hope to minimize any effect of these HABs on aquaculture production. I obviously don't do this on my own. My group has a collection of talented marine scientists, postdocs, grad students, and undergraduate interns. So thanks for listening, and please feel free to reach out to me with any questions or comments through email. My email is jlsmith@fims.edu. Thanks for joining.